speaker today is Paula Cannon. Paula is working to develop therapies for HIV/AIDS patients. She's doing this by going after one gene that she's going to tell us about. She's figured out a way to knock this gene out in mice, and now she's going to knock it out in HIV patients, or at least try. She was trained at the University of Liverpool and at Oxford and at Harvard, and she's now working on this project with the team that she leads at the med school at the University of Southern California. Paula, thank you.
Thank you. So I could not have had a better introduction. Thank you, Jack, because I'm hoping now that your appetite has all been whet a little bit for something that's different than drugs. And I was thinking about how to describe gene therapy. And I suppose I would say that if I was at the Battle of Mobile Bay and I was on a ship and there were a bunch of torpedoes heading at me, I would invent a helicopter. Anyway. Okay. So, full disclosure, I'm looking out at you all, and you probably think you look pretty normal, but to me, I'm a gene therapist. This is what you look like. You are just a bag of genes. Okay, maybe we can be a little bit more sophisticated, but not much. So genes, DNA, they make RNAs, some of them make proteins, some of them regulate how those RNAs make proteins, but at the end of the day, what we end up with is proteins, and that makes something we call a phenotype, and people who aren't scientists would actually call that a body. So this is the sort of, basic thing we're looking at. So if you're a gene therapist, not only is this how you view, healthy people, but your view of disease is also informed by this. And really, my view of disease is genes behaving badly. And that can include genes that you are born with. You're born with a genetic disease, cystic fibrosis, sickle cell disease. Or you could acquire a disease in just one cell in your body. And this is clearly what happens in cancer when a mutation arises, which then leads towards a cancer phenotype. But even in the example of infectious disease, in many cases, I also think of that as a genetic disease, because you are being invaded by a genetic parasite. A very good example of that would be the HIV virus, which not only does it get in your body, but it inserts its own genetic material into the cells it infects, and it becomes a permanent genetic parasite in your body. So if you kind of go along with my gene-based theory of disease, then it also stands to reason that the way we currently treat disease probably isn't adequate. Because the way we treat disease is we try to mask or mitigate the phenotype by sticking chemicals in our body, right? Now, I also work in California, so I also don't like the idea of chemicals. So instead, what the gene therapist does is the gene therapist works in this sort of like different space where we actually think about going to the source of the problem and fixing it. Okay, so the concept of gene therapy is quite straightforward in that you have a gene, a sequence of DNA, and somewhere in that DNA there is an error, there is a mutation, and gene therapy simply needs to fix that mutation and turn it back to what it should be. And that would be great, except for the first kind of 20 years or so we've been doing gene therapy. We haven't really had the capability to do that. And instead what we've sort of done is something a little bit more clumsy. So if you imagine this is a cell, and these are chromosomes, and the little red squiggle is the defective gene, then what gene therapy does is we add in an additional copy of a functioning gene. We often do that by using things like viruses that we hijack to use as gene delivery vehicles. But at the end of the day, what you're left with is kind of, if you like, two copies of a gene, because your added gene does not replace the defective gene. It's kind of like an add-on. Some of the time, that's fine. But there's two real problems, and one is this, is that because our new gene that is supposed to be making up for the defective gene is in a completely different place in the genome, you can get inappropriate levels of gene expression, too much, in the wrong cells, at the wrong time, maybe it's not at therapeutic levels. And in addition, and sort of more sinister, is on some occasions, this addition of a gene can also lead to a process that we call insertional mutagenesis. And that's shown on the little cartoon on the left. No, it's on your right. And what this means is that the gene we have added in, you know what, I'm frightened to use this button. Hold on. This is the one everybody's had trouble with. Okay. The gene that we've added, it can activate a neighboring gene. It can switch on a gene that's nearby and shouldn't be switched on. And if that gene is an oncogene, then you've got a problem. Okay, so the field of gene therapy officially started in September 1990, a clinical trial at NIH on this little girl, Ashanti Da Silva. Well, she was little at the time. I guess she's in her 30s now. And she had a genetic disease called ADA SCID. SCID is severe combined immune deficiency. It basically means there's a genetic problem with your immune system, and as a result, you are susceptible to all sorts of diseases. The form that Ashanti had, ADA SCID, she's missing the ADA gene. It's treatable. There is a drug. It's a stabilized protein form of the ADA protein. You have to inject it every week, and it's hugely expensive, but it was keeping her alive. And so she was the first person to receive gene therapy. And before you ask me, we don't know if it worked. Ashanti got the gene therapy. A viral vector was used to introduce the missing ADA gene into certain cells, T cells, that could be taken out of her body, engineered, and then put back in. But Ashanti's doctors and parents, like good doctors and parents, said, well, okay, this is a first-in-man test, and she's doing pretty well on her current drug, PEG-ADA. So we're kind of okay with her having the gene therapy. What we're not okay with is having her stop taking the PEG-ADA. So to my knowledge, she still takes PEG-ADA. To my knowledge, she's still healthy. But the knowledge we don't have is whether or not that very first gene therapy experiment worked. However, it did galvanize the field. And in the sort of 10 years after this first experiment, we really saw an explosion in the number of clinical trials for gene therapy, up to about 400 a year. And you'll see that they were mostly done for cancer patients, which again is probably to do with the fact that this was still a very new and experimental therapy, so you wanted to be sort of working with patients who, you know, had kind of run out of more conventional options. But despite this sort of early optimism, we certainly had some early setbacks. And one I want to tell you about is one that was reported in 2000, and this is the headline. I actually kept the New York Times from that day. Even though I live in LA, I do read the New York Times. And this was reporting the sort of, you know, unfortunate news that a clinical trial of gene therapy that was going on in Paris that was treating children with another immune deficiency, this time it's called X-linked SCID, that although 13 patients had been treated and 10 of them were actually looking like they'd been cured, what then happened was 1 and ultimately 3 of those patients developed leukemia. And the reason they developed leukemia was because of this problem of insertional mutagenesis, where the therapeutic gene that was put in, here you go, it's called the IL-2 receptor gamma common chain. This, in the millions of cells that were treated, in 1 or even a handful of the cells, it had the misfortune to land next to a known oncogene associated with childhood leukemias, LMO2. And in the 3 patients that went on to develop leukemia, it was all because of clonal expansion of those cells that had this insertional mutagenesis event. So this is the problem with gene therapy in the year 2000 when this happened. And so it really focused people on understanding that if gene therapy was going to be more broadly useful, we needed to go back to this idea of having much more precision in our therapy, of being able to actually reach in and make changes exactly where the genetic mutation is. And so then about sort of in the sort of mid-2000s, a new tool started to become available to gene therapists. And this is going to be the subject of my talk. And this class of molecules are called targeted nucleases. And you may have heard of some of them because, for example, one of these reagents is the CRISPR-Cas9 system that's been getting a lot of publicity in the last couple of years. And I'm also going to be talking about a reagent called zinc finger nucleases, which is the ZFN. And you can basically think of these as genetic scissors. The characteristics of targeted nucleases are they are reagents that we can engineer so that they recognize a specific DNA sequence in a specific gene. And when they get there, they act like genetic scissors, and they cut a hole in the DNA. And that DNA break, which by itself doesn't sound awfully useful, we can actually exploit that and have various different outcomes. One outcome is that the gene can actually be destroyed. Another outcome is that we can go in and make precise changes. I'm going to show you how we do that, but it's based on the idea that the DNA repair process that the cell then starts to do to sort of stick back together the two ends of its DNA can be hijacked by gene therapists to get the outcomes that we want. Okay, so I'm going to give you a little bit of information about zinc finger nucleases so you can understand how these things work. So zinc finger nucleases they have two components, the zinc finger part and the nuclease part. So first of all, the zinc finger part. Zinc fingers are peptides that are components of mammalian DNA transcription factors, so they inherently have DNA binding properties. And what's kind of cool about them is at the tip of the finger, there's about 3 amino acids, which you see here, which make contact with the DNA, and they actually make contact with typically 3 bases of DNA. And by changing the nature of those amino acids at the tip, you can kind of produce a library of different zinc fingers that have different triplets of DNA that they will bind to. Okay? So it's kind of like you make an alphabet. And then you can string together an array of zinc fingers, and you can increase in multiples of 3 the extent of the DNA sequence that you can specifically bind to. And then for the nuclease part, what we do is we add on a nuclease domain. We steal it from a restriction enzyme. And the nuclease domains, they like to dimerize, so they will bring together 2 different halves of your zinc finger nuclease pair that will straddle a longer DNA sequence, and you end up now with a protein-DNA complex that is capable of producing a DNA cut exactly at your target site. So zinc finger nucleases are engineerable and recognize DNA, and they are proteins. There is another example of a targeted nuclease that works in a similar way. I'm just showing it here. It's called a tailin. In this case, the DNA recognition modules come from a bacterial protein, and they only recognize 1 base, so there's only basically 4 different types, a G, an A, a T, and a C recognizing module. But in many ways, they are very similar to the zinc finger nucleases. And then over on the right, we have the CRISPR-Cas9 system, which is similar and different. CRISPR-Cas9 works because the Cas9 protein, which is a nuclease, is brought to its specific target sequence by the action of an associated RNA, the CRISPR. And we call this a guide RNA. And in this guide RNA, you'll see this little region in yellow. This can be changed, and it is made to be homologous to the DNA sequence that you want to target. So it's a much simpler system in a way than sort of having to construct these rather more complicated proteins like the zinc fingers and the tailins. And people do joke that the CRISPR-Cas9 technology has made sort of gene therapy available to high school students. And I can certainly agree with that. I have high school students in my lab, and their project is go make a CRISPR-Cas9 and cure whatever genetic disease you think we should be working on. So it's kind of that's how to get students interested in science, by the way. Right? Okay, so how does what happens? I've told you they make a DNA break. What happens beyond that? Okay, so if you just have something like a zinc finger nuclease sitting on your specific gene of interest and it makes a DNA break, what happens next? Well, one of the things that happens is the cell has various different ways to repair DNA breaks. It has to, otherwise every time we went through an X-ray screening machine at the airport, we would all get cancer. So one of the mechanisms that the cell uses is a pathway called non-homologous end joining. And it's kind of like a Band-Aid repair process. It literally takes the ends of the DNA and stuffs them back together again. And it does it in a very inaccurate way. And what happens is you can lose little bits of your DNA, or you can duplicate bits of your DNA. And so you end up with an insertion or a deletion at the site of the DNA break. And for those of you who know about open reading frames, which is everybody in this audience I know, you can see that it could be relatively easy to use this process and end up with an out-of-frame gene. So that although most of your DNA sequence is still there, you have now effectively lost the function of the gene. Okay, well, knocking out a gene, cool. I don't know, how many diseases can you think of where knocking out the gene would be curative? Because if you can think of some, we can start a company. Because believe you me, there are very few. However, there is one gene that's become sort of like, if you like, the poster child for this technology, and I'm going to tell you about that. And in order to tell you about it, I'm going to sort of sidetrack a little bit first, and we're going to talk about HIV/AIDS. Okay. You guys probably think, I don't know, a lot of you are drug people, aren't you? So you probably think that we actually have a really good treatment for HIV/AIDS in this country. Yeah? Pretty good, hey? Nobody talks about it anymore. There's no public service announcements about HIV/AIDS. I live in Los Angeles. 20% of gay teenagers are HIV positive in Los Angeles. So that's where this lack of messaging gets you, okay? Let me show you the reality in this country. Okay, let me show you this first. Okay, this is how bad it used to be. Many of you are as old or older than me, so you probably remember these scenes from the 1980s. But today, you know, we have drug formulations that are down to one pill, and people take them, and they actually have normal lifespans. So again, it sounds pretty good. But in the United States, this is the reality of antiretroviral therapy for HIV-positive individuals. There's over 1 million people infected with HIV, and it's estimated that only 25% of those people actually have what we would call an optimal outcome. And an optimal outcome means, you know, they go to a doctor, they get diagnosed, they get into care, they are retained in care, they are prescribed an antiretroviral therapy, and importantly, they keep taking it because they don't have side effects they can't handle, they don't have psychological issues around taking one drug every day for the rest of their life, and they don't move around and lose their insurance. So 25% is not really all that great, is it, for 1 million plus people? Okay, even for people who get their art, take their art every day, and have this outcome, it's still not great. So there are side effects of loosely categorized as inflammation. There are diseases that look like accelerated aging. There are toxicities associated with the drugs, and occasionally you will get virus resistance. The drugs are expensive. I don't know, again, some of you might think that's a good thing, but it's estimated that if you're 21 years old and you had put on art and you live to be in your 70s, then the lifetime drug cost will be $500,000. And then, and this to me is the kicker, is that even if you take your art religiously, even if you have the best possible outcome and it drops the levels of virus in your body to undetectable levels, you still have HIV in your body because some HIV just hangs out in kind of a latent state. And that HIV can wake up again if you ever stop taking your drugs. And so because of that, antiretroviral therapy doesn't ever cure people and has to be taken for life. Okay, right. So Mother Nature is smarter than us, and it knows another way to stop HIV. And it's to do with the gene I'm going to be talking about, which is this gene, CCR5. This is a cartoon showing HIV getting into a cell. HIV infects CD4 T cells, and it does so because it makes contact with two cell surface molecules. First of all, it makes contact with the CD4 protein, and that exposes a binding pocket on the fusion protein on the outside of HIV that now allows it to make contact with what is called a co-receptor. And the most common co-receptor that HIV uses is CCR5. Now, what's interesting is you can't live without CD4. It has a pretty important day job in your body. But you can live without CCR5, and indeed about 1% of the population are CCR5 negative, and as a result, they are naturally resistant to HIV. And this is because they have two copies of a gene with the mutation called CCR5 delta 32. It's a deletion of 32 bases, and it basically makes a truncated, non-functional form of CCR5, and these people, therefore, do not get infected by HIV. So, kind of like an early GWAS study. It's kind of interesting, but so what? Well, one of the things that really got gene therapists and me as well kind of excited about this was the report about this man. So Timothy Ray Brown, he's also called the Berlin patient. He's the only person who's ever been cured of HIV. And Timothy was cured of HIV because he had the, I don't know if it's good luck or bad luck, he was living in Berlin. He's actually from Seattle originally. He was living in Berlin. He was HIV positive, and he developed leukemia. And, you know, you may or may not know this, but you know, let's do it real simply. If you have something like leukemia, you're going to be given things like chemotherapy. And if the chemotherapy works, great. You go home, you're cured. But if the chemotherapy doesn't work and we have to kind of like change the regimen, give you more and more doses, what happens is at some point you come up against a sort of a toxicity ceiling where the amount of drug treatment you need to be given, not only will it kill your leukemia, it will also kill cells such as the bone marrow stem cells that you have that give rise to your whole blood and immune system. And if we kill your bone marrow stem cells, you may be cured of your leukemia, but you're not going to live for very long. So one of the things that happens sometimes is people who have leukemia have the drug regimen, and then it's followed up with a bone marrow transplant to sort of replace their own stem cells that have been destroyed during the chemotherapy. So Timothy had leukemia, and he needed a bone marrow transplant as part of his treatment. And his doctor, I love the story, Gero Hutter, his name is, at the Charité Hospital in Berlin. He wasn't even an HIV doc. He was a cancer doc, but he was well-educated and he read a lot. And he'd heard about CCR5 and CCR5 delta 32 and how people who had this 2 copies of this allele didn't get HIV. And so he asked the question, can I find Timothy a bone marrow transplant donor who is not just a tissue match, but is also somebody who is in the kind of 1% of the population who have 2 copies of this allele? Now, Timothy also got lucky, I think, because he was in a country with socialized medicine. Can you imagine, like, trying to do that over here? And so what they were able to do was to go to the database of, bone marrow transplant donors in Germany. I think they had almost 200 people who were potential tissue matches. In Germany, 8% of the population have registered to be donors. And they started doing PCR tests on them. And I think number 65 that they tested was not just a tissue match, but was also somebody who was a delta 32 homozygote. And that person agreed to be Timothy's bone marrow donor. And Timothy had his bone marrow transplantation. And so basically, Timothy was cured of his leukemia and his HIV. In order to understand that, this is just a schematic to remind me to tell you, in case people don't know this, that I'm talking about bone marrow stem cells, which are also called hematopoietic stem cells, or HSC. That's the abbreviation I will be using. They give rise to all the cells of our blood and immune system, including the CD4 positive T cells that HIV infects. And Timothy's treatment kind of looked like this. Here's Timothy. He's been given chemotherapy, radiotherapy. It's wiping out his cancer and his bone marrow stem cells. And so he's been given a transplant from his donor. And if you like, overnight, Timothy went from being CCR5 positive to CCR5 negative. And as a result, he was able to cure himself of his HIV. So you could propose to do that to people who are HIV positive. You could say, why don't we find everybody who's HIV positive a matched bone marrow transplant donor who's CCR5 negative and do a transplant? Not a stupid question, except that bone marrow transplantations have a very high rate of morbidity and mortality, about 30%. You would not wish them on your worst enemy unless your worst enemy was going to die of leukemia. So it's not a trivial thing to do. But instead, what we want to try and do with gene therapy is recapitulate what Timothy had, but this time to do it with the patient's own hematopoietic stem cells. Take them out, engineer them in the lab with our targeted nucleases, and knock out the CCR5 gene to make at least a portion of those cells CCR5 negative and then return them to the patient. Okay, so this was the idea, and this was the work plan. First of all, we have to figure out how to do it efficiently in the hematopoietic stem cells. We have to make sure we don't do any harm, we don't damage the stem cells while we're doing it. And then, importantly, we have to show that this actually works to suppress HIV. This is for those of you who like technologies. I work a lot with a biotech company as a partner, Sangamo Biosciences, and together we sort of figured out that the best way to do this was to actually have messenger RNAs that coded for the zinc finger nucleases to electroporate hematopoietic stem cells so that the RNA goes in and transiently expresses the zinc finger nucleases, the genetic scissors. And there's a machine that allows us to do a patient-sized dose, which is typically about 200 million cells. And the efficiency we're getting right now, we're knocking out about 50% of the CCR5 alleles. One of the things I was asked to do was to develop an animal model to look at this. So again, now I'm feeling very defensive about animal models, so I have to go carefully here. But the animal model that we work with to test out this therapy are called humanized mice. And humanized mice are basically mice that receive a mini human bone marrow transplant. We put some of the human hematopoietic stem cells into them, and because these mice are themselves immune deficient, they accept the human cells, and they will grow as many of the components of a human immune system, including here's human CD4 positive T cells in their blood. And because they have these cells, of course, they can then be infected by HIV, and we can test out our therapy. This is just a slide. We did an awful lot of safety data, but this is showing that if you have mice that receive either unmodified or zinc finger nuclease knockout hematopoietic stem cells, and you follow them over time, the quality and the type of the human immune system that develops in the mice is indistinguishable, suggesting that we haven't done any harm to the hematopoietic potential of our stem cells. But then I suppose most importantly is this slide. This is the slide to show you that this therapy can work to suppress HIV. So again, what we have here is mice that received either control, untreated human hematopoietic stem cells, or the ZFN-treated ones, and we infected them with HIV. And you'll notice that early on in the first few weeks, all of the mice got infected with HIV. So our therapy did not stop our mice getting infected. And that kind of makes sense to me, because remember, dirty little secret about gene therapists, we're not 100% efficient. So probably the majority of the cells I put into the mice were not modified, were still CCR5 positive, and only a minority was CCR5 negative. However, over time, what happened is the mice were able to control their HIV levels. And at the same time as they did that, when we looked at the levels of human CD4 T cells in their blood, you'll see that although the control mice, the levels crashed very quickly because HIV is killing those cells, in our ZFN-treated mice, although we had an initial dip, the T cells then picked up again and recovered. And that's natural selection at work. HIV, if you like, was kind of part of its own demise because it's selected for the cells that were resistant. And in addition, if we take some of these cells that persisted and were selected for and grew out in the mice, and we look at them, we can actually see evidence that the CCR5 gene was indeed damaged. Here's some sort of sequencing data where we're sort of sequencing around the site where the zinc fingers cut. And we see all these deletions, and they're very pleiotropic. And these are classical signatures of non-homologous end joining repair. Remember, just sticking it back together against some insertions and some deletions. So the zinc finger nucleases are working as we expected, and in the presence of HIV, the CCR5 negative cells are the ones that persist, and over time, the levels of HIV were suppressed. So people like this idea. This is probably the highlight of my career so far. I found this in an airport. I never read this magazine. But in 2011, we were the #2 science story of the year. I know you, oh, thank you. So, okay, I always get asked, and I can never remember what the #1 story was. I think it was the Higgs boson. It was something physicsy, okay? So I think this says something, doesn't it? People like this idea because it makes sense. It's elegant and it's simple, okay? Well, all right. But we've still got to translate it. Everything I've shown you, beautiful as it is, has been done in a mouse. And so we had to figure out how to scale this up to the size of a human. And in order to do that, it costs a lot of money. But we were lucky, okay? CERM, which is the California Stem Cell Agency, was the Proposition 71 that the good citizens of the Republic of California voted into law in 2004. Thank you, George Bush, for making embryonic stem cell research illegal and making us do this. Sorry, is that a bit political for this song? But this really gave us the opportunity to go to this funding agency and say, we have a stem cell-based therapy, and we want to apply it to this disease of HIV/AIDS. They allowed us to form what's called a disease team, and it was a fantastic collaboration. My team at the University of Southern California, colleagues at City of Hope, which is a big cancer hospital with expertise in cell processing and bone marrow transplant, also in Los Angeles. And then we had a biotech partner, Sangamo Biosciences, that are up in the Bay Area. And this is just sort of showing you really where we are with moving this into the clinic. We started in 2010 with our first chunk of money from CIRM. They gave us $14.5 million in that first round of funding. We got IND approval in 2015. CIRM then kicked in with a second round of funding to actually allow us to do the clinical trials, and we've just enrolled our first and treated our first patients in the last month. So we are on course to do a trial. Larry, you will like this because it's a clinical trial. Because it's gene therapy, the numbers involved are 3 plus 3 plus 3. So it's gene therapy, so we do things small scale. This is the actual details of the trial. I put this slide in just so that if people are watching it on the web and they want to look it up, that's the reference on clinicaltrials.gov. I have an easier to understand picture of it. Basically, patient, you want to go back? Oh, okay. Okay, who are the population? So the population, it's a very broad age range. They are people actually who are doing okay. Usually, one of the ways when we first thought about who the appropriate patient population should be, and I'm going to consider this my question time now, okay? Right? You start off thinking, okay, who are the patients we should do? Well, our first thought was, let's have AIDS patients who need a bone marrow transplant. Let's have people who have AIDS lymphoma. Awesome, because that's a perfect experiment. Unfortunately, frontline therapy for lymphoma has got really good, and very few AIDS patients with lymphoma progress to needing a transplant, so those patients just don't exist. So then we're like, damn. Okay, let's have patients who are taking antiretroviral therapy but are doing really badly on it. So their virus is suppressed, but their CD4 T cells haven't come back up to normal levels. And let's pick an arbitrary number of, I don't know, below 500 CD4 counts, which is sort of these numbers. Again, there's not many of those patients around. So at some point, you have to kind of say, well, do you want to do a clinical trial or not? Well, we do want to do a clinical trial. So this is the sort of criteria we've now come up with. And that's kind of not a bad range. These are reasonably healthy people. We have 3 different sites, and people are, signing up for this. We'll come to this in a moment about why they're doing this. So again, here's my cartoon. This is showing a patient. First of all, we have to give the patient a drug that will mobilize the hematopoietic stem cells out of their bone marrow because, I don't know, people don't like having big needles put in their bone marrow and their stem cells being pulled out. People much prefer to have the stem cells, at least temporarily, be kind of present in their blood where we can purify them. We then treat them with zinc finger nucleases by electroporation of the messenger RNA, and then we return the stem cells to the patient. And an important point to note here is that we don't just infuse the stem cells back into the patient because stem cells get lost and they don't know they have to go back to the bone marrow. So what you have to do is you have to give the patient what we call conditioning, a drug such as busulfan, which is kind of like chemotherapy light. You do a little bit of damage to the bone marrow niche so that the new stem cells you've put back in will actually home back to the bone marrow. So that's kind of an unknown in this trial. We don't actually know what the optimal dose is of busulfan, and so part of the escalation in the trial is to increase the dosage of busulfan to improve this sort of re-engraftment. So to summarize, and I've got another story after this, but not very long. To summarize, we're using targeted nucleases such as zinc finger nucleases to disrupt specific genes. It's providing a new way to treat HIV by knocking out CCR5. We're a team of academics, clinicians, and a biotech partner, and we were lucky we lived in California. And I want to say, you know what? The antiretroviral therapy drugs are not the Beatles. So even though they sound okay, people do still want something new. We've had a lot of support from patients who say to us, we need something better than these drugs and lifelong drug therapy. And of course, our ultimate goal is that because these are stem cells and they should last for life, that this could potentially be a one-time treatment that could lead to remission for HIV.
So will you take them off drugs?
Yes, yes. Okay, also, noticing this is also part of my question time. Yes, we will keep them on antiretroviral therapy for the first 12 months after they receive the cells, and then as long as they are doing okay on everything, and we have evidence that we can see the CCR5 negative cells. Yes, and that's what we will do. So we will have a planned treatment interruption of 6 weeks to take them off the drugs. We'll monitor them very carefully because what will happen is the virus will come roaring back. That's our expectation. And so we will kind of monitor the patients and work with the clinicians. And then our hope is, it would be awesome if the virus didn't come roaring back, or even if the virus came roaring back and then was suppressed, like we saw in the mice. But even, this is a phase 1 trial, so even if we just see that we can see some evidence for selection of increased levels of CCR5 negative cells during this treatment interruption, that would be considered good as well, right? All right, so in the last couple of minutes, I want to tell you about what else we can do with targeted nucleases beyond HIV, and specifically beyond knocking out genes. Because remember, most of the diseases we're interested in, diseases of the blood and immune system, knocking out the gene's not going to help it. If you've got sickle cell disease and we knock out the beta globin gene, well, you're going to turn people from sickle cell disease into beta thalassemia, which is much worse. So, we need to be able to go in and correct genes, not just knock them out. And so in order to do that, we need to look at a whole different way of doing DNA repair. So far I've told you that a DNA break can be repaired by the non-homologous end joining pathway, and that can lead to gene disruption. But there's another repair pathway in the cell, which is much more elegant, which is called homologous recombination repair. And homologous recombination repair is basically what happens if you have a defect in one chromosome. The cell will basically say, I don't know, what should that be? Let me look at the other chromosome. Let me bring it over here and use it as a template to repair the defective chromosome. So that's homologous recombination repair. And we can kind of exploit that pathway because if we introduce what we call a homology donor, which can be a plasmid, a DNA template, or a viral vector, as long as it has homology to the DNA sequence, so this would, for example, be a stretch of CCR5, maybe a kilobase of the CCR5 gene, the cell can then instead copy our introduced homology donor. And this gives us an opportunity in our introduced homology donor to make the change that we would like to see, to make the correction for the sickle cell genetic change. And then we can end up with what we really think of as mutation correction. In addition, as well as just having like one little base change here, we can also open up our homology donor and insert something into the middle of it, so that this also gives us a capability to have targeted site-specific insertion of a gene. Remember the problem of insertional mutagenesis? Well, this is one way to get around it by having this very specific gene addition. And in the final 2 slides I'm going to show you with data, I'm going to show you how we know this is working, and our sort of surrogate for this is that I'm going to site-specifically insert a GFP reporter as a surrogate for this process. So one of the things we discovered recently is that AAV vectors, which is a small DNA virus, work really well as homology donors. So in the viral genome, we put our flanking homology arms, in this case CCR5, and our correction or our GFP reporter. AAV is also kind of like the M&M of viruses. It comes in many different flavors and serotypes, and you can find one that will work for your cells of interest. So AAV subtype 6, for example, works really well with hematopoietic stem cells. So we basically combine the two. And this is just showing you data that happens when I use AAV donors containing homology arms or not, with and without the matched zinc finger nucleases. And over here on the right is the data showing that only in the case when I have the matched reagents do I get a high level of site-specific insertion, in this case of GFP, that gives me long-lasting expression of GFP, in this case in 30% of my hematopoietic stem cells. Oh, this is just showing you that if I take those cells and put them into my humanized mouse model, and then I look at the blood in the mice at about 4 months, you can see that each of these, this is 4 individual mice, and they all have on average levels of GFP expression that matches quite closely to what the input cells had going in. Okay, so I talked about this earlier, but I'm going to end on this point. So X-linked SCID, which is this horrible genetic disease where there's a defect in the IL-2 receptor common gamma chain. And some of you may recognize what this photograph is. This is the little boy David, the boy in the bubble, who was a child who was born with X-linked SCID. He lived for 13 years entirely in a bubble to keep him sterile and safe and away from all the germs that his immune system couldn't cope with. And this was also the disease that halted the gene therapy trials that were going so well in Paris back in 2002. So I would say, one of the first disease targets for this sort of precise gene correction is absolutely X-linked SCID, because instead of adding an extra copy of the gamma C chain, instead what we'd like to do is we can go in and we can make zinc finger nucleases and homology donors that match to the site of mutations in individual patients. And I just leave you with one piece of data that wasn't even optimized, just showing that we can indeed make changes at this specific locus. Okay, so to summarize, I've shown you that targeted nucleases, such as zinc finger nucleases, can be used to knock out a gene, and we're actually quite a long way along in that process now, and we have that now in clinical trials. But in addition, by combining them with homology donors based on AAV6 vectors, we can now start to get gene correction in addition. And although all the data I've shown you today has been with zinc finger nucleases, this works just as well when you combine the AAV6 donors with CRISPR-Cas9 as your genetic scissors. And then the efficiency of our gene editing capability is now really approaching that you get with standard gene therapy vectors, but of course with much more precision. And I'll just finish with a slide thanking members of my lab, past and present, collaborators, and sources of funding. Thank you.
Before we take a break, let's take a couple of more questions for Paula.
I think you should
This one's about CCR5. In order for 1% of the population to be a homozygous for one particular mutation like
Right.
that, for one allele, there must have been a dreadful plague
Right.
that these people are the progeny of the survivors.
Why
Anybody
do
know
we
what it
have it?
was?
Yes, we do. So the question is, why do we have CCR5 negative people? People used to think it could have been bubonic plague because everything gets blamed on, bubonic plague. But actually, molecular clock analysis has shown it happened much more recently, and the assumption now is that it's to do with smallpox. It's not an experiment we can test, but we believe that people who are CCR5 negative are probably less likely to die when they have smallpox, because CCR5 is to do with regulating your immune response.
Yes, there's a question from a woman about 2/3 of the way up in the center in brown.
Is there an immune response to the virus when you try to edit the cells? Wasn't there another step, like a clinical trial, where there's someone who was, I think, heterozygous, and they didn't die from this disease that was usually fatal, and
Right.
they tried to edit his genes, and he ended up having...
Sure. So you're thinking about the sad case of Jesse Gelsinger, who had OTC deficiency and died as a result of a massive immune response to adenovirus vectors that were infused into his liver to deliver a therapeutic gene. So gene therapists always worry about immune responses against our vectors, against our transgenes. To some extent, the applications I've been talking about are mitigated because we take the cells out of the body, and especially with the zinc finger nucleases, we're kind of using RNA to just transiently express the zinc finger nucleases, and then it goes away. They make the cut and then they're gone. So it's kind of hit and run gene therapy. And even with the AAV vectors, the field understands quite well now how to monitor for and prevent or mitigate if any immune response does happen. So certainly people, we're watching for that, but so far we haven't seen it.
Take one more question. Yes, over here in the front. A microphone's on its way. Yes. Yeah.
Have other states copied California?
Oh, yes. Don't they always? Yes. Yeah, New York does. Gosh, there are others. I mean, obviously, I'm just going to say, this is a bloody great idea for California. I mean, not just in terms of how awesome this is going to be for humankind when 10 years from now we're treating everything with gene therapy and stem cell therapy and not chemicals, but it's really, it was such a validation of science in California. And, we were able to persuade all the best and brightest stem cell scientists from around the world to come to California. It's an awesome place to be a stem cell scientist. So I think it's a great model. I think, oh my God, I should call up Donald Trump, shouldn't I? So I think states should do things independently and not always rely on NIH to do things.
I had the pleasure of hearing Sir Elton John speak about his foundation work in 2009 at the Bio Conference. Is Sir Elton John aware of your work? Do you know?
Who's that, sorry?
Sir Elton John, the musician. His foundation funds a lot of AIDS research.
Elton John. Sorry, it was the Sir bit.
But he is a Sir.
I think he is. I've done quite a few charity events for HIV/AIDS.
Prolific in his fundraising and his foundation.
Yeah, one of the issues I will say we run into, I'm just going to say it because everybody asks me, it's like, oh yeah, this is cute, but how are you going to roll this out in sub-Saharan Africa? So there's still a little bit of tension between, we're trying to do something that's kind of cutting edge and maybe in the future, and I honestly believe this is where medicine is going because it's going to work and it's elegant. We just need to figure out the scale-up and production aspects of it. But there's still a little tension because I am working in a disease where, we're doing a clinical trial with 9 people in Los Angeles and Connecticut and San Francisco, and of course the vast majority of the disease burden is not in those places. So limited resources, huge amount of money still needs to go for vaccine development and all the other, more vanilla things we should do, yeah. But there's always a bit of a tension around that. So.
Paula, thank you so much.
Okay.